I'm delighted Dr. Goss could be here to speak to us. He is a professor of medicine at the University of Washington and the director of the Cystic Fibrosis Therapeutics Development Network. I think most of you know the Therapeutics Development Network is the overreaching umbrella that organizes and supports clinical trials for therapeutics in cystic fibrosis. You might not have known that Johns Hopkins is one of the initial 7 centers in the TDN, which was 25 years old now.
Yes.
And is now over 90 centers. So I'm delighted that he can speak to us today about cystic fibrosis and the new therapies and research that are going on right now. So thank you.
Great. And I want to thank Peter for inviting me and also the whole team at Johns Hopkins. We were just remembering that actually Noah Lecton and I got our first R01 together, big NIH grant in 2003 or 4. And together we studied home spirometry in CF. But it's been a very tight relationship with the University of Washington and Johns Hopkins. We've done a lot of collaborative studies together, and we still do. It's actually great. It's been a great relationship. But here I am to talk to you about a little bit about the CF therapeutic pipeline. And we created this slide here a while ago to look at all the therapies that have come with organized structure of clinical research. So this is the predicted survival on the y-axis, and then the year on the x-axis. And it doesn't go all the way to 2023, but what you'll see is the different therapies and then when they came in. But for a rare disease with about 40,000 patients, to have this kind of therapeutic development is really extraordinary. And it happened because of this sort of, the CF Foundation has the wisdom to have care centers, and then on top of those care centers, create research centers to actually facilitate clinical trials for novel therapeutics. So again, the new median survival, this is 2022, is 56.6 years. And that's very different from when I started in CF clinic in 1998. So this is the pipeline. I love this slide. It's very busy, but that's what you want. It just shows, this was, I think Bob Bell, the prior president of the CF Foundation, was smart enough to realize you're going to have hits and losses. But this pipeline is dynamic, it changes, and it just tells you how many therapeutic agents are being evaluated even now in the post-Trikafta era of CF. And it gives you the phase of development of the drug, but also the different areas of drug development. And what you'll see is the big blue area there on the left, which is really looking at more basic treatments to fix to repair the protein. So this is the TDN. Just to give you a sense of the size of the blue dots are the actual centers and how big the centers are. And you see Hopkins is a nice big center out there. We're up there in the corner in Seattle. But this is now, again, 92 sites around the country. So almost every patient can actually get to a site fairly easily, except for if you live in this sort of more the West where we are, where some patients have to travel a long ways for clinical trials. But there really is a clinical trial near almost everybody with CF in the country. And I think that's really a great thing for CF. There are other centers here. I just say this is the Therapeutic Development Network in blue. There are more centers. This is the Success with Therapies, another sort of form of sort of improving sort of patient-centered outcomes. This is the NTM centers. So again, often they're the same centers starting to treat non-tuberculous mycobacterium CF. And this is the learning network, where you're trying to use quality improvement to improve the care of CF patients. But many of these sites will be the same, and this is all supported by the CF Foundation. And the last is the lung transplant consortium, knowing that we have to improve the outcomes of transplant for CF. So again, it's the extent of the support of these sort of networks is really extraordinary and unlike any other disease. And it comes, here's the research funding for 2022, $225 million. And I want to focus, in this last year, they supported almost $60 million in genetic therapies and treatments, nonsense and rare mutations. That is one of the big struggles we still have after Trikafta. 10% of patients will not be on Trikafta. Some people who are eligible for Trikafta can't tolerate it due to either hepatotoxicity or liver toxicity or other intolerances. So it's really important that we advance that arm of therapeutics. So this is, I think, the path to the cure, I think, is really visionary. The CF Foundation committed $500 million to the path to the cure. And this is really three big strategies: repairing broken CFTR, restoring CFTR protein when none exists, thinking like stop mutations, fixing or replacing underlying genetic mutations, and all really get at the root cause. And I think the hope is that there'll be a single therapy at a single time point, and your CF will be cured. This will include new modulators, mRNA therapy, and gene therapy and gene editing. So this is the sort of the four big strategies. You get gene therapy with gene editing. Then there's nonsense, where you actually read through a nonsense, which is a stop mutation. The protein coding tells it to stop prematurely on the protein, and then also mRNA or small nucleotides. And then protein therapy, which is really CFTR modulators. That's where we spent the last almost 15 years and led to such great advances in CF. And then there's also, we can't forget the CF manifestations. Patients still have symptoms, those both on Trikafta and those not on Trikafta. So standard therapies and traditional therapies need to be advanced. So really, the approach to treating the basic defect includes sort of getting better potentiators. These are drugs that open up CFTR channels like ivacaftor. Correctors, this is part of the Trikafta combination, which help this defective CFTR move to the surface of the cell. And then read-through components that allow you to read through a protein. And then I think the real future right now is in RNA therapy. So this is ways to either just give the correct RNA to the cells so they can start making protein, much like our COVID vaccines many of us have received over the last 4 years. And then gene editing techniques and repair to fix the basic defect. And really, this is an area that has exploded in the last 5 years. And how's it exploded? Well, genetic-based therapies are really a reality. I started in CF, and we were studying genetic therapies. We were in Seattle. There was a company called Targeted Genetics. They had an AAV, an adeno-associated virus inside was packaged the CFTR. And after discovering the gene, they thought it'd be 10 years to actually get a gene-based therapy. That all went to the wayside. It sort of quieted down. The study didn't work. And then then the CF Foundation pivoted and started to look on small molecules. And that was very fortuitous because that actually generated Trikafta. But now things are changed. I think the environment is so different. Sickle cell treatments, now using CRISPR-Cas9 gene editing are successful. 2 drugs are approved, FDA approved, and this is a simple technique. While you take peripheral blood out, you find early red blood cells, you edit them, and then you put them back in, and then they repopulate the bone marrow of these patients, and then their sickling stops in their blood. And it's really a dramatic improvement. And now these are just some examples. These are all FDA-approved gene therapies. And again, I think this is going to be the era of gene therapy in the next 10 years for CF. And CF has done the CF Foundation has a very thoughtful approach to thinking about gene therapy. They have this inverted pyramid, I think, sort of tells you how they're doing it. So there's a huge focus on preclinical development, drug discovery, drug formulation, pharmacology, and toxicology. And then in the middle there, then we have an alignment with this CF Foundation, the TDN. We try to figure out who's ready to move forward into humans. And then we have phase 1, 2, and 3, and 4 clinical trials. Those are the different phases of clinical trials for drug development. Phase 4 is after approval, where we move into the clinical human arena. And I think that this vision, I think, was quite remarkable. And as before the creation of ivacaftor, this is using a very similar approach. the CF Foundation provided money, and they're getting all these different technologies and platforms. And then from that, we start to look at the laboratory proof of concepts, some preclinical development, so early phase, and then selection optimization of a particular gene therapy. Then it'll move in the TDN, and then it will go to patients. And then from there, hopefully we get a new therapy. And again, what's different about CF is this movement into humans is very thoughtful and careful and emphasizing safety. So there's a collaboration with the CF Foundation and the TDN Coordinating Center where I am, industry, and then back again. And there are lots of back and forth until we see that the protocol is appropriate, safety measures are in place. Patients are part of this process. Trey sees a lot of these as part of our clinical research executive committee. Again, we have 2 patients. We have patients on the data safety monitoring board. We have patients on our protocol review committee to look and say, is it a feasible? It may be the protocol may be what the company wants, but if a patient says this is not doable, they need to hear that. And we get that sort of constant feedback. So it does take a while, but then hopefully we get the best product that goes into clinical trials. And I think they have all the components for success. So they have early regulatory engagement. The CF Foundation will help with proactive and targeted advocacy for regulatory agencies. The coordinating center, we really partner with the CF Foundation to help figure out ways to engage companies and make sure the protocols make sense. There's academic industry partners. The CF Foundation, probably the best foundation in the country, and some would say the world, in how they've leveraged pharmaceutical company relationships to develop drugs that actually go to patients and change their lives. And then there's early development. We created early guidance for companies to how to get engaged and active in creating a drug for CF. There's a CF lab resources. So the CF lab can de-risk a lot of these early products. They have many, many different cell lines of mutations that you can test your product in early. And then there's animal model infrastructure. So we have the CF ferret, we have CF pigs, which are very expensive. There's a CF rat at UAB. These are model systems that can help think about how you're going to develop a drug. And again, what has that turned into? Well, there are now 7 active and 7 engaged companies in gene addition, 7 active and 6 engaged in gene editing. Vehicle delivery. So it's actually, how do you package that DNA or mRNA? So there's 7 active companies for vehicle delivery, 7 engaged, and then read through or splice mutations. These are sort of things that can correct these stop mutations. There are 4 active programs. And then we're creating models and tools to help make sure we get the right product and move it forward. So these are some of the mRNA therapies that are ongoing. Arcturus, Vertex, and Recode is about to start. These are in phase 1 trials. I think Hopkins is a site for Vertex's trial. And then Splicense is it's a oligonucleotide. Then 4D is now in phase 1 trial, almost finishing its phase 1 trial. And then Spirovant is entering. So these are basically companies that are really either in the clinic or about to go in the clinic. And as you sort of say, it's not a one shot on goal kind of thing. It's many shots on goal. And if you look at these are all the preclinical companies that are moving into CF gene therapy. That's a lot of companies. So they're taking a very thoughtful and really aggressive approach to thinking about this, and including really investing in really novel technology. So these are the gene delivery. So when you're going to send a gene and get it into a human, you have different ways of doing it. Adeno-associated viruses are viruses you can package the DNA in. Then there are lipid nanoparticles. These have all been used in the past. And then there's a human Boca virus, another kind of virus. But they're doing all these other technologies that didn't exist when we started doing gene therapy back in 2000. So they have polymers that can package the DNA. There's cell penetrating and cell specific peptides that can actually deliver the cargo. Extracellular vesicles. These are just things that can actually very different from viruses. And then there are phage derived particles. Phages are little particles that are usually they're phages can be directed against antibiotics, but you can also direct them against human cells too and get your DNA in. And there are also a lot of approaches to thinking about full length CFTR gene insertion. How do you insert a gene, the corrected gene? And the first one is utilizing RNA guides, so Cas9 or zinc fingers. This is to retarget recombinases. You get it to the right location. Then you can what's called reprogram the recombinases. You can utilize what's called directed evolution, or site-specific mutagenesis to alter the DNA targeting. And what I'm really interested in is this last group, which is actually companies that utilize what's called prime editing. They figure out a way to put a unique landing pad on your DNA, so you only insert the DNA in the correct spot. These are all things that are coming, will be probably an evolution over the next 5 years. So how are we going to optimize all this and actually conduct this? And this is where a new trial that's about to start is going to help us. We need to optimize what's called trial design. Gene therapy trials that actually go to market often are quite small. They're not large trials, even in phase 3. But again, there are not that many. There are only 10% of the CF population are not on modulators. So you're getting a smaller and smaller population to target. How do you do that without only having one drug? How do you move multiple products through? And the idea was to create a control data and use endpoints, and then other companies can leverage whatever their treatment arm is and then use more of these control spots so you don't have as many people on placebo. So there are different sources of control data that can help you say, what is it like if you didn't get the gene therapy? And for that, we've designed something called the REACH study. This is a study that will complement control data. We'll talk about that a little bit later, but you can take data from past investigator-initiated studies that'll tell you what is the natural history of not getting a gene therapy and following clear clinical protocols for lung function to set intervals. We also have data so we can create subsets within the CF registry data to look at that natural history. And last, actually, for some industry they've given the data from trials to the coordinating center, so we can actually use that to simulate what it'd be like if you didn't get a drug, to try to understand the safety of a drug. And last, you can create what's called the REACH prospective study, which is about to start. And this is REACH. It's a cohort of for patients who are not on CFTR modulators, who are not on Trikafta. And what we'll do is we'll follow them for 12 months. These little houses represent where there'll be home spirometry, and we'll collect symptoms. The big stars are when they're going to get office spirometry and do a patient-reported outcome measure, a questionnaire. And then they'll also be getting blood collection at specific times. And the whole idea is to let these patients can go in and out of this study and actually participate in a trial, go back in. But this data can then be used to augment placebo arms of clinical trials. And this makes doing a study much more efficient in a small population. But I'd have to say, I'm very excited about gene therapy. I never thought I'd see Trikafta, to be very honest. When they got ivacaftor, I said, oh, that was great. How would they ever get that's an easy problem. You have ivacaftor works because it attaches to a protein that's on the surface and it activates it. That's a very common model in drug development. But Trikafta is a combination drug where you actually move the mutant protein to the surface and activate it. That's very, very challenging. So I was very skeptical whether we'd ever see this day, but I'm actually not skeptical that we'll see a gene therapy for CF before I retire. And I don't intend to retire that soon, but in the next 10 years, I think we'll probably have a genetic-based therapy for CF. I'd be very surprised if we didn't. But I think they're not stopping there. There's still important challenges for CF. And I think one area that they're working on, which I find we've been involved in a couple of these trials, which is bacteriophages. And these we're filled with bacteriophages. They're little viruses against bacteria, and they're very specific. They don't attach to human cells, only to bacteria. And you can actually either design them, or you can actually collect them from the environment and specifically collect ones that attack Pseudomonas or other problematic pathogens in CF. So what they do is they attach the bacteria, they insert their genetic material into the bacteria. The bacteria then start generating more of the phages. They rupture, and the phages then go on and infect the other bacteria. And there are two major industry trials that are in phase 2, Biomics and Armada, both hopefully reporting soon about their results. We were in the Armada trial. And the CFF is also starting a study to support phage-specific therapy for Mycobacterium abscessus, a very difficult bug to treat. And this is a study that's going to be called Post-Stamps. I think it's going to be opening soon. We're trying to be a site. I think we are a site. And it's basically where you take the abscessus, you send it to the University of Pittsburgh. They test different bacteriophages against it, and then they will give it, then they'll send all this information back to us, and we'll start bacteriophage. They'll send us the bacteriophage, and we'll give it to patients with this problematic infection. There are a lot of other funded studies, multi-center studies that the CF Foundation supports. So one is, these are some of the studies that are investigator-initiated. They've been really important in actually helping us understand different aspects of CF care. And I'll be only talking about a couple of them, but these are purely CF Foundation. There are no companies involved. These are investigator-initiated studies, and they've led to a better understanding of a lot of aspects of CF. I'll make special mention of two studies. One is Mayflower, which is studying pregnant women with CF. So most of us in the adult clinic have just shown an explosion of pregnancies. Our standard Monday clinic is a little, I think we need a nursery, because it's like there are so many little kids running around. And there were, I think the number of CF mothers is now well over 600. It has stayed that way since Trikafta came. And it's really an extraordinary change in CF as itself. And my first paper in CF was in pregnancy. So coming back to it now and watching this boon and having all mothers in these families be, having, actually, I have one patient, she has like 4 or 5 children now. So it's just a very different world than it was when I came into CF. And the other one with special mention, I think, is the bionic pancreas. This is a study where you have a single device that measures your glucose and gives you insulin. And, a lot of our adults have diabetes. And so they have there's constant, in the old days of diabetes, is finger sticks all day long, adjusting your insulin all day long, very challenging for, it's almost like one of my patients said it was worse than CF, like getting diabetes was worse than CF, harder to deal with. But this is a device that you'll put on. It'll do everything at once. It'll go to the cloud. It'll use AI to predict your insulin needs, and it'll hopefully really improve that glucose balance over time. Now, I did want to sort of give you some highlights of, I think, some of the big advances we've had. And this is a study I just still revel in. This is the PROMISE study. PROMISE studied, the CF Foundation started this study in about 3 months. And that's when Trikafta was getting evaluated, when it was sent to the FDA. It was approved in 6 weeks from the FDA. It was just incredibly rapid approval. This study was stood up and activated. And what we were trying to do is, for all the patients who were in the trial, we wanted to see, did the results replicate what the trial was going to do? And then we wanted to measure some other aspects of that Vertex was not particularly interested in. So this is the change in lung function. This is based on whether you had no modulator before, was a blue line. This is lung function in the far left. And then the middle line is if you were on tezacaftor, ivacaftor, or lumacaftor, and the last if you were just on ivacaftor alone. But it basically replicated the clinical trial. Marked rapid increase in lung function, marked rapid drop in your sweat chloride. And again, most of the patients on the ivacaftor had already had a fall in their sweat chloride. Most of the patients are having sweat chlorides outside of below the diagnostic range for CF now. But this was just an extraordinary. But most recently, and this is a paper that was just published last year. This is about microbiology. What happens in microbiology? So they carefully looked at sputum in these patients. And this is what happened to Staph aureus and Pseudomonas right after starting Trikafta. And the red line is the average of all the patients. So classically, CF patients will have 10 to the 5th, 10 to the 6th density of Pseudomonas and Staph in their sputum, very dense organisms. And then as soon as you start Trikafta, the red line drops very precipitously to 1 month after it, and it stays there. Unfortunately, the vast majority of patients continued with their same pathogen, but the level of Pseudomonas, the level of Staph aureus dropped precipitously. And I'll take a question. It was broad range. The question was, what was the inclusion criteria for PROMISE? It was all comers. However, they didn't get many people in the severe group. So people between 40 and 70. So the majority of lung function was actually pretty good. And I also wanted to highlight one other study that's going and rolling. And this is close to my heart. This is called the STOP 360, and it's called this is the aminoglycoside study. So our current treatment for exacerbations, the guidelines suggest if you have Pseudomonas, you should be on 2 anti-Pseudomonal agents. And the most commonly thing we use something called a beta-lactam, and then we use generally aminoglycoside. That's almost always tobramycin. There are a lot of prospective trials and retrospective analyses that may be suggesting that the addition of these 2 agents may not be as beneficial as we used to think. In old CF, when I started, I really ignored the toxicity of tobramycin. I mean, patients were very sick. I needed to do whatever I could to get their lung function back. Tobramycin will induce what's called vestibular toxicity. You'll lose your balance. It will cause ototoxicity. You'll have loss of high-frequency hearing, and you may have kidney injury. But we accepted a little bit of that toxicity because my argument was that the need was great. But is that still important now? Kids, do they need these 2 agents? I've had patients who've had probably 30 or 40 courses of IV tobramycin. So this study was designed to say, is that still beneficial? But I want to say who are the key leaders. I hope you recognize Natalie West. She's not here. I told her I'd show her a picture. So she's one of the key leaders here in the study. It's myself, Patrick Flume from MUSC, Sonia Helsey's our lead statistician, and Natalie. And Natalie's been instrumental for our STOP program just from the beginning, studying exacerbation. So this study, these are the sites. We actually have some more sites here. There's sites all over the country. There'll be over 700 patients who are experiencing exacerbation. We randomized to either get an aminoglycoside and a beta-lactam or a beta-lactam alone to see if it's still beneficial to use tobramycin. And we have some additional data from our last study to say maybe it's not that useful. This is data from our prior large-scale randomized controlled trial comparing duration of therapy, but it's how people did in lung function and symptoms after initiating antibiotics. And so this is 1 anti-Pseudomonal, 2 class of anti-Pseudomonal, and 3+. And what it shows is they all have a fairly similar improvement in lung function and a similar decrease in symptoms. So maybe more is not necessarily better, but we need to figure that out. And so this is the study. We're going to compare aminoglycoside plus beta-lactam versus beta-lactam alone. And it's a randomized controlled trial. And it's open label. You'll know what you're getting. And the planned IV course is about 14 days plus or minus 2. And then we use study-specific guidelines to dictate what antibiotics you get. But it's basically made by clinicians to make sure that they have the choices of the antibiotics, but they fall in these categories. And this is what it's going to look like, or what it does look like. It's ongoing. So you get randomized, then you get your active intervention, and then the follow-up period. But we've learned from patients that actually the less study visits you have, the better. Make it easier to do. Make it something that's sort of part of care. So they're seen at visit 1 and visit 2, and then that's 28 days later, and then we have a phone call at visit 3, which is about 44 days later. So it's a very simple study to enroll in. And this is our enrollment graph. So the black line is where we stand. This is the number of sites we have. I think the newest site we have almost 60 sites are up and going. So we're ahead of enrollment. We've already enrolled 102 patients, and again, we're trying to get this over 700 patients in the end. So we'll have an answer hopefully in about 2 years whether the toxicity is warranted, whether actually adding this agent is helpful. Again, it's been our dogma. We have done this ever since I have been taking care of adults with CF and kids. And again, what I want to say it's interesting is the CF Foundation is really willing to sort of reject dogma and go back and test it again. And this is, I think this is particularly relevant for children because I'm not sure that this added toxicity early on is that beneficial. We need to show that it's beneficial. So. But we are off to the races. But I want thank you for your attention. But I just wanted to give you just a snippet of what's going on, and then what you can anticipate in the next decade of CF. Again, I'm very optimistic. I think the CF Foundation and the whole network they've created, the scientific resources to advance therapies is extraordinary. I mean, there's nothing like it. And there's actually nothing like it in adult medicine. No other disease in adult medicine other than HIV and maybe Hep C have seen the kind of benefits that we've seen in CF. Certainly in pulmonary clinic, it's the only clinic where you really see marked change in outcomes. So stark that we actually now have to move all of our fellows into clinic because they don't see patients enough in the inpatient setting. When I was a fellow, I never saw a patient in clinic. I only saw patients in the hospital. That's a very big transition. And we rarely have more than one patient in the hospital now, where we always had 5, 10, 15 patients in the hospital. That's a pretty different disease. So I think, for patients, there are still those 10% that are not on Trikafta. I think it's really critical we solve this puzzle for them. Not doing that would be a disservice to the community. And I think, it's the right thing to do. So we have to keep developing new therapies, even for traditional problems like infection and inflammation. But we also have to go shoot for the cure because I think it's really going to be within our future. So, but with that, I'll take any questions from the ethosphere or here. So.
Thanks. What are there efforts being made to apply artificial intelligence or deep learning to either look at large patient data sets to figure out correlations or predictions to help with outcomes and treatment regimens? Is it also being used maybe to optimize research investments? I'm just curious what's going on in this space.
Yeah, the AI space, and I can't speak to all the AI space. I'm not really an expert in AI, but I know that there are some efforts to look at the registry data that almost 90% of all CF patients provide their data to get insights from that. I know there have been machine learning approaches to trying to look at prognosis. And they do what's interesting is there's a big emphasis on trying to combine different sources of data, and then potentially using AI to understand it. The British have a big AI program going right now out of the Royal Brompton. I'm not sure where it stands. I just know that they had started it.
I just wanted to say Dr. Vanskoy is the lead on our Precision Center of Excellence at Hopkins, and they are working on culling the EPIC data to try to enhance that with other metrics as well. To answer exactly that question, can you figure out best therapeutic strategies, predictions for who's going to get diabetes or other issues as well in the clinical? But it's early. There's early game.
And I do think AI is being used when looking at sort of disease pathways or metagenomic data, genomic data. I know that's ongoing to try to get better insights into potential pathways that have not been targeted or drug targeted for CF.
What do you think the next therapy that we're going to see that's going to come out out of this?
Yeah, no, if I had a crystal ball and I would say the next likely successful therapy we'll see in this space is probably going to be an mRNA therapy. I think it's mechanistically, it just makes sense. We've all received a lot of mRNA. It seems to work. I mean, the COVID vaccine basically took mRNA therapeutics and exploded it. So I think it's whether that therapy needs, one would argue that you're probably going to have to get it multiple times, like we do get the COVID vaccine multiple times. But just, so the potential to actually, biologically, the lung is a different target than your arm. And whether you can get through mucus and whether you get proteolysis or things gets degenerated. But I think it just makes intuitive sense that it's not a big stretch to come from a COVID vaccine to a mRNA-based therapy for CFTR. I think gene editing is a lot further away.
There's an online question about research about preventing diabetes and anything about lung transplant advances.
Yeah. So, and I don't know all the treatments for preventing diabetes. I think that's a really important question. Even understanding CF-related diabetes is important. It's, there are translational studies looking at pancreases, CF pancreases. This is from biorepositories trying to understand what is the mechanism by which CF patients are at such high risk of diabetes. Is it all what we call innocent bystander, meaning the exocrine pancreas, the thing that makes our digestive enzymes, causing this environment where the islet cells are failing, but not anything specific for CF? Or is there other reasons that they can actually salvage that? I think that their islet cell transplant is also a topic that I know people are talking about. It hasn't happened to any great degree in CF. For transplant, I'll pivot to transplant because transplant has had a very different stake for CF. So the number of transplant patients with CF on the list right now, or people awaiting transplant, has fallen dramatically with Trikafta. I think it's now about 30 patients. I think I just saw the data last week. I think it's about 30 patients this year actively in the United States, in the entire United States. And it basically, after Trikafta, the numbers just plummeted and it stayed low. But there's a transplant consortium to try to improve outcomes, and they are trying to prevent something called chronic lung allograft rejection, which is the limiting step in transplant, the longevity. But it's important to note that CF is not, lung transplant, the outcomes have not improved that much. In CF, they're actually very good. So one of my patients, he had his lung transplant in 1991. We celebrated his retirement. That's a long-standing organ. And so CF has they have the best survival of anybody after transplant. The average survival in Canada, I think, is 12 or 13 years. That's still not good enough. So hopefully they're also, I think that Lung Transplant Consortium is about to launch new studies in better immunomodulatory drugs. So how do you prevent the host from slowly rejecting your lung? And I think that they also have a giant biorepository. I think they have 400 patients in this biorepository where CF patients who've had a lung transplant are giving their blood, their cells from their bronchoalveolar lavage, and it's being collected and stored. It's at Cleveland Clinics right now. That's the coordinating center. And they're trying to understand the immunology of rejection. And they're including actually non-CF patients in that giant study. But I think it's an interesting site. So actually non-CF patients are going to benefit from the CF's effort to improve outcomes in transplant. But hopefully transplant will have that same evolution that other organ transplants like liver transplant have done, and kidney transplant, which outcomes are really quite good. So.
Thank you, Dr. Goss. On the mRNA therapeutic, what does that mean for people who are, people who are on modulator be eligible for that?
Yeah, so the mRNA therapy current trials, people who are on Trikafta or are not eligible, although some of the studies are changing actually. So there may be later cohorts may be allowed to be entered. But it's an important question. I think they're primarily focused on these non-modulator eligible patients. But that is changing. And again, genetic-based therapies are usually small. They have a lot of sites just to allow enrollment. But again, like you can always sort of follow the inclusion criteria because that's going to be changing. But right now, I think the mRNA studies I've seen have not yet included, but it may include patients who were modulator eligible, but not on it because of a toxicity. So that has changed. And that was input from CF patients and providers. And then, but whether you're actually on Trikafta and maybe you didn't think you got his optimal response, still be.
Hi. It's great to think big, and I think the success of the TDN, is really a force. But is it possible to take these studies internationally and globally?
Yeah, and I didn't really show that. So a good example is for investigator-initiated studies, we've done a couple. One was something called SHIP, where it was a pediatric study looking at CAT scan and hypertonic saline. That was had European centers, and actually the Stop 360 is adding 11 sites in Europe and England. And then the genetic-based therapies are almost all going to be trans globe. It does pose challenges because the regulatory bodies are very different, and to get them aligned is challenging. So, but to really advance a genetic-based therapy, it's going to take a global perspective. The CF-TDN Coordinating Center and the CTN, the Clinical Trial Network in Europe, meet regularly to discuss that. And there's also the UK network to discuss how we're going to sort of move forward with these. There are some gene therapies that are starting in Europe or starting in Australia and will be coming to the United States in time. So.
Are there any studies looking at changes to like nutritional guidelines or BMI guidelines for people who are on the modulators?
Yeah, there's a important new study. It was called CF Strong, which is actually looking at nutrition in the setting of modulators. There's some suggestion. People are worried that Trikafta increases your weight, but it may not be good weight. May not be lean body mass. And that's if you're and actually better understanding that. And so that study is I think that study is about to start. And I think Hopkins is a site, I believe. But it is an important thing to understand. I think the guidelines are going to be changing. Our nutritionist now basically advocates a Mediterranean diet, more standard for anyone on Trikafta. It's basically as Trey was noting, a lot of our patients, my winner in my clinic, he ate 5,000 calories a day and he couldn't gain a pound. That's extraordinary. That's the same nutritional input of the linebackers, the defensive line at the UW Huskies. They eat that much. And now on Trikafta, he cannot eat 5,000 calories and not get a pound. So it is going to be changing. I think the guidance is not yet out, but maybe there's new word.
So there was a position paper out just this past year from the foundation, but there's no data to say definitively that, we recommend this, but the main idea was that everything should be individualized because everybody's nutritional response to Trikafta is different. Some people gain a lot of weight, some people don't. And the idea is that if people are growing well or maintaining well, that it should be more like the healthy diet for the general population. And if people are having trouble gaining weight, then it would be more of the CF legacy diet. So in clinic, we're trying to implement that, but it's sometimes hard because we've been telling people for 20 years, eat. And all of a sudden we're like, hey, maybe we should back. So we're trying to work more and do a little more like anticipatory guidance. And hopefully there'll be more data soon. Oh, and we just got a bioelectrical impedance analysis machine, which measures lean body mass in clinic. So on the PED side, we got a grant, and so that we can track not just weight, but lean body mass, because Dr. Goss was talking about what kind of weight are you gaining, and so that we can say to kids, let's get more muscle. Like we're not talking about weight, just let's get stronger. We can work with physical therapy and use the bioelectrical impedance machine. We just got it, Izzy and I just got it up and running. So.
Again, but it's a great question. I think we're going to see very different, it's already changed. I mean, we've changed our tone in clinic much to this personalized tone for each patient. So. Thank you very much.